In this experiment, we studied microevolution in real time by tracking how E. coli populations evolve resistance to the antibiotic ampicillin. So to start off, we began by creating a mixed population of ampicillin-resistant and ampicillin-sensitive E. coli, and then we grew the bacteria under two conditions, with and without ampicillin. And then each week, we transferred a small volume of culture to fresh media while making sure to maintain selective pressure in the ampicillin-treated group. Over multiple generations, we expected to observe evolutionary change driven by natural selection, while it was favoring resistant strains in the presence of the antibiotic. E. coli is a common, fast-growing bacterium used in lab studies, and ampicillin is a beta-lactam antibiotic that kills bacteria by interfering with their ability to form cell walls. Some E. coli carry a gene that makes them resistant, usually on a plasmid, which ultimately encodes an enzyme called beta-lactamase. And this enzyme breaks down ampicillin before it can harm the bacteria. And so for our experiment, these resistant strains had a survival advantage in environments containing ampicillin. We predicted that selective pressure from ampicillin would lead to more resistant bacteria over time. So in cultures grown in LB without ampicillin, both resistant and sensitive bacteria would survive. Therefore, we expect to see a high number of colonies on both LB and LB plus ampicillin plates, but fewer on the AMP plate since only resistant cells will grow. And then in cultures grown in LB plus AMP, only resistant bacteria should survive and replicate. So we expect both the LB and LB plus ampicillin plates to show similar and high colony counts. And when plating bacteria from an LB only culture, we predict that there will end up being more colonies on LB plates and fewer on LB plus ampicillin plates due to the presence of both sensitive and resistant strains. And so for our culture grown in the lab with no AMP, when we do the culture swab with the plate with no ampicillin, it should be around 100 colonies because both resistant and non-resistant grow freely. And then on the flip side, for the culture with the AMP, it should be 0 colonies because no ampicillin present to test the resistance. And then for the cultures grown in lab plus ampicillin, for the no AMP colonies, it should be 0 colonies because this condition doesn't exist here since the ampicillin is present. And then for the ampicillin culture, there should be around a couple hundred because only ampicillin-resistant bacteria survive here.